Hi guys, today I will be talking about hemoglobin. Hemoglobin is an extremely important iron-containing metalloprotein which can be found in red blood cells we call erythrocytes. Thanks to hemoglobin, the oxygen we breathe is transported to tissues through our bloodstream. Tissues need oxygen to perform their metabolic activity. In particular, oxygen is necessary in a set of processes called aerobic respiration. During this process, the ATP, adenosine triphosphate, is created, which is the major energy storage unit in our body. When we take a look at our red blood cells, hemoglobin makes up about 95% of their dry mass, which is quite a lot. In mammals, one hemoglobin can carry up to 4 oxygen molecules. Besides its great role in transporting oxygen, hemoglobin is also responsible for carbon dioxide transport. Carbon dioxide is generated by metabolic processes in our body and we have to get rid of it. We do this partly by breathing the gas out. Hemoglobin is also the reason why our blood is red. Let's talk about hemoglobin structure for a while. Hemoglobin has 4 subunits, as you can see in the picture here, so we say that it is a tetramer. If we look at the normal adult hemoglobin, we have 2 alpha polypeptide chains and 2 beta polypeptide chains. Both alpha and beta chains have almost similar length, but of course they differ in their amino acid content and order. Other types of hemoglobin exist, for example fetal hemoglobin, which has 2 alpha chains and 2 gamma chains, or sickle cell hemoglobin, which has 2 alpha chains and 2 modified beta chains. Alpha chains in these forms are always the same. Now you can see the structure of the heme. Heme is a prosthetic group of hemoglobin. It is an iron-containing cyclic tetrapyrrole. One heme group can bind 1 molecule of oxygen. As you can see in the picture, the ferrous form of iron is in the center of tetrapyrrole. 4 molecules of pyrroles are linked by methane bridges. Because there are conjugated double bonds everywhere, heme absorbs visible light and has deep red color. There are several substituents present all around the molecule. You can see methyl in blue color, vinyl in green color, and propionate in orange color. If the center iron atom is oxidized from ferrous form to ferritic form, the biological activity is destroyed and heme cannot bind oxygen molecule. Now, let's take a closer look at oxygen transport. The important term here is PO2, which is partial pressure of oxygen in our bloodstream. The unit, which is mostly used in medicine for this purpose, is milliliter of mercury, which approximately equals 1 torr. Historically, pressure of gases was firstly measured accurately by mercury manometers. They are of course less used today because of mercury toxicity. Hemoglobin behaves as if it were 2 different proteins based on oxygen levels. When partial pressure of oxygen is high, let's say above 100 milliliters of mercury, that is, there is a lot of oxygen molecules present, so this is happening in the lungs, hemoglobin displays a high affinity for oxygen that enables it to bind oxygen to nearly every available heme ion. This form of hemoglobin is called the R state, relaxed state. On the other hand, when partial pressure of oxygen is low, below 40 milliliters of mercury, so this is happening in tissues, hemoglobin has lower affinity to oxygen. Transition of hemoglobin to this low affinity, which is called tau or T state, enables it to release a large proportion of the oxygen previously picked up in the lungs. This dynamic interchange between the R and T state serves as the foundation for hemoglobin's sigmoidal oxygen binding curve, which you can see in the picture here. On the x-axis there is partial pressure of oxygen, on the y-axis there is percent of saturation by oxygen. In the violet color we have oxygenated blood from lungs that goes through arteries to pass the oxygen to tissues. In the dark pink color, we have reduced blood which travels through veins from tissues into the lungs to be oxidized again. It is important to note that T and R states have different conformations. Now it is time to talk about carbon dioxide transport. Carbon dioxide is generated in peripheral tissues by Krebs cycle. Carbon dioxide immediately combines with water to form weak carbonic acid, which dissociates into protons and bicarbonate ions. Deoxyhemoglobin, T-state hemoglobin, acts as a buffer against acidification by binding these protons and delivering them to the lungs. It can bind 2 protons per tetramer. So, we say that hemoglobin has lower affinity to oxygen in more acidic environment, that is tissues, when compared to more neutral environment of lungs. In tissues, oxygen is therefore easily released. In the lungs, as oxygen binds to deoxyhemoglobin, the resulting transition to the R state triggers release of these bound protons. The liberated protons combine with bicarbonate to form carbonic acid. This acid is dehydrated during a process catalyzed by the enzyme carbonic anhydrase to form carbon dioxide, which is then exhaled. This coupling of the interconversion of carbon dioxide and carbonic acid with the binding of protons and oxygen by T and R state hemoglobin is termed the Bohr effect. In tissues, decrease of pH in red blood cells promotes the synthesis of 2,3-biphosphoglycerate. This compound can bind through salt bridges to hemoglobin in T state to its central cavity. It therefore stabilizes deoxygenated hemoglobin because there are now additional salt bridges that must be broken down prior to conversion into R state. In fetal hemoglobin, biphosphoglycerate binds weaklier. There is 1 salt bridge missing compared to adult hemoglobin, and therefore there is lower stabilization of T state, which means that fetal hemoglobin has higher affinity for oxygen than adult hemoglobin. Let's talk about methemoglobinemia. In this, it is a blood disorder in which an abnormal amount of methemoglobin is produced. In methemoglobin, ferrous iron is oxidated to ferritic iron. Normally, enzyme called methemoglobin reductase helps in iron reduction. One can have oxidated iron in hemoglobin as a side effect of agents such as sulfonamides, or activity of methemoglobin reductase is lowered. Another case is inheritance of the gene for a mutationally altered form of hemoglobin called hemoglobin M. In hemoglobin M, histidine has been replaced by tyrosine. In alpha-chain hemoglobin M, oxygen affinity is lowered and there is no Bohr effect present. Another type of mutated hemoglobin is hemoglobin S, where the nonpolar amino acid valine has replaced the polar surface residue of glutamic acid in the 6th position of beta subunit, generating a hydrophobic sticky patch on the surface of the beta subunit of both oxyhemoglobin and deoxyhemoglobin. In tissues where the partial pressure of oxygen is lowered, deoxyhemoglobin can polymerize to form long insoluble fibers, as you can see in the picture. Because of these fibers, erythrocytes have characteristic sickle shape. And lastly, I will mention so-called glycated hemoglobin. Blood glucose that enters erythrocytes can form a covalent adduct with part of hemoglobin beta chain. This process is called glycation and it is not enzyme catalyzed. In healthy adults, this glycated hemoglobin represents around 5% and it is proportionate to blood glucose concentration. Because erythrocytes have half-life around 60 days, glycated hemoglobin reflects the information about mean blood glucose concentrations over the several weeks. This information is therefore useful in management of diabetes mellitus. So that was all from me about hemoglobin. I hope you did find it at least a bit useful, and thank you for watching and see you next time. Bye!